I want to introduce our very own speaker today. I'm sure you all know him. So we have Dr. Aurelien De Jode. I practice this. And he is, in case you don't know, he's a postdoctoral researcher at the Titus lab and also affiliated with the University of Alabama. And he's going to talk about marine snails today. I hope you're excited and please take it away.
Thank you, Kat. Can you hear me? Yeah. Yeah, so today I'm going to talk to you about the work or part of the work I did during my previous postdoc at the University of Gothenburg. And so this is going to be mostly about genomics, speciation, and those snails that you can see here on the picture. So a few general word on speciation first. So this is a complex evolutionary process that goes from one population. So we can start here. And that goes to two different species that are fully isolated. So I'm going to use a few terms here like barrier to gene flow or reproductive isolation. So for reproductive isolation, what I mean by that is the reduction in interbreeding between the populations and also the reduction in gene exchange between populations. And so if we look at this small graph here, you can see that you can have actually different levels of reproductive isolation on what we are calling the speciation continuum. So the idea is that like you can accumulate reproductive isolation a long time, and this can be gradual or sudden, or it can have different shape. And in the end, you end up with two fully isolated populations. You can also see here that is a representation of the genomic differentiation between the two populations along the process. So at first, you only have like a few genes that are barriers to the gene flow. And you can see this type of like peak high differentiation between population. But as the process goes on, the whole genome is going to be resistant to the exchange of genes. And so you're going to have higher and higher differentiation all along the genome. So now this is, we are interested in speciation. So we see this as in a direction, but really it doesn't have to go in any direction. It can start, it can stay stalled somewhere here. It can go back depending on an important number of factors. And so because also there is a lot of different components in the process, some people have proposed to look at it in a more multidimensional way. So this is just one example of a speciation cube, if you wish. So you have, for example, you take into account the genetic divergence, the phenotypic divergence, and the reproductive isolation, which is like a more integrative way to look at the process. Now, often we have access to this, potentially to this, but direct measures of reproductive isolations and other factors is a bit more tricky in like natural systems. So one thing that is like kind of easy to understand, which is it's the appearance of the first component of reproductive isolation, because you can think about it like just, I mean, in nature, most populations have some type of, or kind of, to a certain extent, locally adapted to the environment. So this is already a first component of reproductive isolation. But then what is a bit more tricky to understand is how the other components of this isolation appear and goes together or like coming right after the other until you get two fully isolated species. So like one of the factor obviously is like time, because you need time for example, selection to act and produce a barrier to gene flow. You also need time if you want to develop some level of genetic incompatibilities between the population. You can also have along the process a stronger divergent selection that applies. For example, if you have the habitats are becoming more distinct, and that's also going to increase the level of reproductive isolation. You can have spatial arrangements and demographic history that play a role into this, because they are more conducive to the appearance of reproductive isolation components. So you can see that, for example, if you in a configuration where you have gene flow, this is supposed to oppose divergence. But if you see uh, a secondary contact, which is a contact between population after they've been geographically isolated, it's like an opportunity for gene flow, but it's also an opportunity for the appearance of reinforcement, which can lead to higher reproductive isolation. Then you can have the evolution also of assortative mating, which is the reproduction with individuals that are more alike. And that's obviously, if it appears in the two populations, that's going to create an extra barrier to gene exchange. And this can be realized through behavior or through habitat choice, for example. And then you have also the genomic architecture of your system that plays an important role. So like what are the genetic architecture of the traits that are under divergent selection? So is it like one loci that have a strong effect, several loci distributed along the genome, and how your patterns of recombination, which includes also genomic rearrangements, and we're going to talk about that a lot in the case of the snails. So one thing that is kind of tricky about this is that we typically cannot observe the full process of speciation, because it's typically taken too many generations for us to observe it directly. So a way to try to go around this in natural system is to try to find pairs of populations that are located at different level of divergence, at different level of reproductive isolation along the speciation continuum. And then you can try to look at those systems and try to study their like phenotypic divergence, genomic divergence, and so on. So this has actually been like, this type of study has been done only in a few systems, and we need to learn more about that, like by adding systems. So like one of the systems is the sticklebacks, right? So where you have the repeated appearance of like freshwater and saltwater ecotypes that are adapted to marine versus freshwater environment. And this is a repeated evolution of the ecotypes that uses mostly standing genetic variation. Another very interesting system is the Heliconius butterfly, where the different clades, for example, Erato and Malpomene, they use different host plants, and they differ in several like traits. And so and they all have like different levels of isolation between them, but also some introgression. And those differences are also linked to genomic rearrangements. And then another example in insects is the Timena insects. So those differ in their host plant. They have host plants ecotype. And so in this system, you have divergent selection on the color pattern. And you also have some type of behavior on the mate choices using the cuticular hydrocarbons of those insects. So those are like system where we've been able to do that type of study. So now to the system of interest. So this is Littorina saxatilis. So those are, sorry, those are different ecotypes of the same snail. So this is a gastropod. It is one of the few that has an ovoviviparous reproduction. So this is pretty cool because actually when you crack a shell open, you can see like the small snails developing in the brood pouch. And those are just tiny little snails that will come out when they are ready. So it is found on the northern Atlantic shore. So from Portugal here, you find them along the French coast, up in Norway, Sweden, the UK. There is actually a weird population in, so there is an invasive population in San Francisco, and there is also a weird population in Venice, in Italy. So this is stuff that are studying now. And so what's very interesting about those snails is like it has repeatedly produced those crab and wave ecotype along the shores of Europe here. So so this is those are the same scale. So this is the snail that's found only in Greenland, I think. This one is called, is only found, in like empty barnacle shells. Those are the two ecotypes found in Sweden, and those are the two ecotypes found in Iberia, in Spain particularly. So typically what you have is like you have a wave ecotype, so this and this, that is like adapted to be resistant to wave action. And you have a crab ecotype that's usually bigger with a thicker shell that is adapted to resist the crab predation. And so, yes, so this is, here you can see a tree about the different populations of Saxatilis. So this is the Spanish one here. So we're going to talk a lot about this one. And then you have all the UK, Ireland, France. But the US is here too, and Iceland, and Norway, Russia. And this is the Swedish population here. This is actually, so Sweden is the place where it has been the most studied, the ecotypes of Lictorana saxatilis, and we're going to compare it to Spain in this study. Yes, and so there have been some demographic modeling done on that species, and it showed that the the ecotypes they evolved in parallel in multiple countries, in all those places. Okay, so this is time for the video, because I think it's a good. So this is just a video that's gonna explain to you a bit the system in Sweden where it's been the most studied. So I think it should work.
Have you ever wondered where the enormous diversity of animals and plants come from, or how one species diverges to form two new species? The answer is speciation. Speciation is the splitting process that generates biological diversity. How does speciation happen? Although not yet fully understood, rough periwinkles, Littorina saxatilis, show us how speciation might happen. These marine snails live in two distinct, contrasting habitats. The rocky headland, where cliffs are battered by the action of the waves, and the boulder shore, home to many predatory crabs. Like any other species, individual rough periwinkles vary a lot. Their shells range from thin and small to thick and large. Some snails spend more time inside their shell. Others prefer to crawl on their foot and grip the rocks. Those with large, thick shells spend longer inside their shells. These crab snails fare better in boulder habitats. Crabs struggle to predate on them. Small wave snails, who like to stick to rocks and hide in crevices, do well on the cliffs, staying firmly attached despite the crashing waves. Through specific traits being favoured by natural selection in each environment, populations can diverge in different parts of the shore. Crab snails in the wave environment are not well adapted to this environment and are less likely to survive and reproduce. The same is true for wave snails in the crab environment. Those with beneficial traits will survive and reproduce, passing on their characteristics to the next generation. This is natural selection. Mating between crab and wave snails opposes local adaptation. Called hybridization, this occurs at habitat boundaries, where the two populations meet. It's costly because it produces poorly adapted intermediate offspring. It slows down speciation. A mixture of traits makes the snails vulnerable in either environment. They are poorly adapted. Hybridization can be avoided if individuals preferentially mate with their own kind. This is known as assortative mating. Another way to avoid hybridization is habitat choice. Snails may evolve to move towards their preferred environment, even if they're born somewhere unsuitable. That is, providing they get there before it's too late. Will these populations evolve into separate species? All the effects we've looked at help to move them in that direction. In some populations, they work together, and speciation may be completed.
So I can put that in the full screen now, I think. Yeah. Okay, so that was a brief description of the system, how it is in Sweden, and I'm gonna complement that a bit. So as you saw in the video, we have so this is actually the place where the video was taken place, as you see. So this is the boulder field where you find the crab ecotype, and you find the wave ecotype on those pretty sharp cliff. But in Sweden, one thing that's important is that it's roughly the same shore level. So there is not much of a tide or anything like that. So regarding this variable, it's pretty much the same for both ecotype. So they differ in different traits, as they say, so shell thickness, shell shape, and behavior. And so there's been a study about phenotypic lines. And you can see that when you go from one end to the transect to the other end, you have a change in size and you have a change in shape of the shells. And then, so those are the different traits that have been measured. So weight, length, thickness. Those are actually gross parameters that are derived from the shell shape using a mathematical model. The coloration we also used, and the bold score. So this, it's basically just a simple test to see how bold the snail is. You just disturb the snail, so it retracts in its shell, and then you wait until and you measure how much time it takes for the snail to get out of the shell again. And so typically what you see is that the wave ecotype is bolder than the crab ecotype, because it's an advantage to be able to get back on your foot quickly if you're in a place that's battered by the waves. So now from a genetic perspective, so again, it's been shown that in Sweden that several loci follow a cline pattern along the transect, which is a strong indicator of the divergent selection that is happening there. In terms of genome, so Litorina saxatilis has a genome that was quite fragmented, but thanks to a linkage map, it has been organized into 17 linkage groups that are the same as the number of chromosomes, which is good. But also, there are several genomic inversions that have been detected on that genome on almost every chromosome, as you can see here. So the gray, so this represents a chromosome, and the gray spots represent the coordinates of genomic inversions that are actually segregating differentially between the crab and the wave ecotype. And finally, so there's been studies on shape and size and other phenotypic traits such as aperture that showed that some inversions are actually implicated in the determinism of those traits. Okay, so this is the Swedish system. So now if we look at the whole picture, I mean, not the whole, but like several spots in Europe. So Sweden, the UK, France, and Spain. There has been a pool sex study. So using pool sequencing to look at the differentiation between ecotypes on those different locations. And so what is what got out of that study is like most likely like the inversions that were found in Sweden are present in the other locations, and also that there is quite some parallelism in the system in the sense that the same regions of the genome that showed high differentiation in Sweden also showed high differentiation in the other countries. And again, another important thing, because we're going to talk about Spain and Sweden later. So as I said, in Sweden, there's not much of a high shore, low shore difference, but in Spain, the gradient from crab to wave is actually the same as the high shore, low shore gradients, which is potentially an explanation for the difference between those two locations. Okay, so now we're just gonna, as I said, just have a like systematic comparison between two transects, one in Spain, one in Sweden. We're gonna use like low coverage whole genome sequencing, and we're also gonna use some phenotypical data to look at to compare the divergence pattern between those two places. So a bit of the sampling, how it was done. So this is the transect in Sweden. So it went from crab to wave. It was 96 snails. And in Spain, as you can see here, it was actually 2 transects. But we pulled those together because there was no differences due to seasonality in our analysis. So we just have all those 2 transects are pulled together. As you can see, like the low shore is only accessible in Spain. It's only accessible during very low tides. And we have like almost 200 snails sequenced there. Actually, there was way more snail sampled, but this is the number of snails that was sequenced in that study. So a bit about the method. So the phenotyping, the boldness I already explained. So this is here the wave snail, typical wave snail. This is the typical crab snail. So for the shell shape, we are actually using this type of picture, and you can place a few like points on the shell, and then somebody in the team developed a computer program that derives from this a model of the shell and calculates some gross parameter that we can later on analyze and discriminate between the two ecotypes. And then we looked also at things like bending patterns. So here you can see that those snails have bends, those have ridges, for example, but they don't all have these characteristics. And then for the genotyping, we use like a whole genome, a low coverage whole genome sequencing. So this is just the bioinformatic pipeline, really. So we had a few tricky spots there, but I'm just gonna go quickly. So basically, we used the random. We subsampled the reads randomly to call the SNPs, and we used 2 different data sets because when we wanted to look at the whole genome and like what happened along the genome, we used all the SNPs we got. But when we wanted to do like more general whole genome average pattern, we just use an unlinked data set by selecting only 1 SNPs every 1 KB window. So now the first results. So those are based on whole genome, the whole genome average whole genome sequencing. So okay, so this is a PCA. So this is Spain and this is Sweden. This is a PCA for whole genome data. So here what you can see is the difference between the two, right? So in Sweden, we have a kind of a continuous genetic variation along the transects from crab to wave, while we're in Spain, we have a very different type of genetic data where there is 2 clear distinct clusters. that correspond to the two ecotypes. Now we can look at this pattern also along the shore. So if you go from a transect from crab to wave, you can see that this is this is the crab ecotype here up there. This is the first component of the PCA here. And you can see that actually what's interesting is like in the crab ecotype, you can see kind of a downward slope towards more wave-like genetic backgrounds. And in Sweden, we find the Klein pattern that we've found in many studies before. So those are the two differences. And also, like if you look on average, the FST is higher in Spain compared to Sweden. Yeah, I talked about the Kleinel pattern in this. And then, so another way to look at this is really the hybrid index. So if you look here in Spain, what you can see is that this is mixed. So sorry. So you can have like two distinct distribution, while in Sweden you have something that is more contiguous. So like this is the first differences on the whole genome level. So now what I'm going to do is I'm going to zoom like chromosome by chromosome, and I'm just going to look at one chromosome. I took this chromosome 17 or linkage group 17 because it's kind of this one got the best figures I have, so that's why. So if you look at the top graph, so sorry, the y-axis here is a genetic differentiation. It goes from 0, so no differentiation, to 1, completely different allele frequency. And here is the same, but in Spain. So typically what you find is like the differentiation is typically is higher on average in Spain compared to Sweden. That's like a generic thing that we found along all the chromosomes. And the other thing that we found is that the genome regions that are known for inversions, they also show up in Spain. So this region here is a region that has inversions in the Swedish system. And you can see here you have a block of very high differentiation between the ecotypes, which is what you expect in case you have a genomic inversion there. So this is actually consistent with the presence of those inversions in Sweden, and it's also the difference between the differentiation in those inversions versus the background is higher in Spain compared to Sweden. Okay. So yeah, I just wanted to tell a bit about genomic inversion in general before I continue. So those are mutations that happen during the meiosis process. And so I just put that as an example to show how it can happen during the meiosis, but really what matters for us at this stage is to realize that we go from this to this, or from this to this, right? So you have these inversions here of the order of the genetic sequence. And what it does is like when you have both those haplotype together in an heterokaryotype, then it prevents recombination, particularly at the breakpoints. And so in a way, it can protect combination of co-adapted alleles from the effect of gene flow and recombination, which can be a very advantageous for local adaptation and speciation. And so genomic inversions have actually been found to play a role in many systems in local adaptation and speciation. And now I just wanted to say also a small thing about how we find them and how we detect them. So of course we could sequence and see the sequence being reversed, but we cannot always do that because of the limitation of the system, the sequencing technologies and that kind of things. But one way to look at it is to take this chromosome 17 here and to do just a PCA on the genotypes like we did for the whole genome, but just on this chromosome or precisely on each of the portion of the chromosome. So this is the portion where you don't have an inversion. So you see it looks like that. It's a PCA between ecotypes. And this is the again, the PCA between the, sorry, not between ecotype, it's the PCA for all the individuals. And so in the regions where you have an inversion, what you see is you can see actually 3 clusters. So the 2 extreme clusters are the individuals that are homozygote, either for the original arrangement or for the inverted arrangement. So you have one homozygote here and the other one here. And the individuals that are in the middle here, they are the individuals carrying the heterozygote. So they have both the original, the ancestral, and the inverted arrangement. So that's how we're going to look at those in our system using this PCA approach. So this is again chromosome 17. So this is the PCA approach on the portion that is inverted. So this is in Spain, this is in Sweden. So the first thing that you can see is that, yeah, we definitely confirm that there is the same inversion in Spain and Sweden. We can, so the colors here, they correspond to the ecotypes. So the red is the crab ecotype and the blue is the wave ecotype. So that's the first thing. And then we wanted to look at those 2 systems together to know if like the same arrangements were present in higher frequency in the same ecotype, which would indicate some sort of parallelism between the systems. And so to do that, we projected the Swedish individuals into the Spanish space on the PCA. And so what you can see here is like, so those here, so this is the Spanish individuals, those are the Swedish individuals, those are the ones that are homozygote for one arrangement, those are the ones that are homozygote for the other arrangements, and this is all the heterokaryotypes here. And so you can see that if you look at the frequency of each arrangement in the different ecotype, you can see that both in Spain and Sweden, the same arrangement is in higher frequency in the wave ecotype compared to the crab ecotype. So this is something I computed later on. So just to show you the frequencies here. So in Spain, you have the difference in frequency for that arrangement between wave and crab, and this is in Sweden. So you have the same type of pattern, but again, the difference is stronger in Sweden. In Spain. And another thing that you can do once you have done that is like looking into only the Spain crab ecotype here. You can look at the proportion of the different arrangements along the transect. And so what you can see is that this this arrangement is the frequency of the arrangements varies along the transect, and this is consistent with what we saw also on the whole genome pattern, where like some the crab ecotype look more like wave-like when going down the shore. Okay, so now another interesting, very interesting thing that we found in that study system is, so now I'm changing chromosome, sorry. So it's about the sex determination. So this is chromosome 14 in the Spanish system, and I'm looking at this specific region, 14.3, and this is chromosome 12 here in Sweden, and I'm looking at this specific arrangements, this region, sorry, 12.2. So we already knew that there was that this chromosome was implicating in sex determination in Sweden, as you can see here, and particularly in the crab ecotype, sorry. So as you can see here, if you look at this, so the snails that are in that have this arrangement here are almost all males, all males, and here you have like 26 female for 5 males. So you have kind of an association depending on between sex and the arrangement you are carrying. So this is an example of like, so the sex determination is genetic in those snails, but it's really not clear. Like it's because it's not 100%, so it's not a fully understood system yet. But when we looked in Spain, we didn't find the same pattern on the chromosome 12, but we found something kind of interesting and confusing on chromosome 14, because we have a similar pattern in the sense that in one ecotype, we have those different arrangements that are clearly associated with sex, but it's actually found in the wave ecotype and not in the crab ecotype, where you have a strong association with sex. So that's like a different sex determinism system between the ecotypes in two locations. And there's people are working on that right now. I cannot say much more about this, but it's a very interesting system. Okay, so now I'm just gonna say a few words about the phenotypes. So the first graph here is actually a PCA that integrates like several phenotypic traits, so the weight, the thickness, and then several of the growth parameters that were measured by that software Shell Shaper. And I've also included some pictures of the extreme shells, if you wish. So typically the first axis is kind of correlated with the size and the weight and the size of the snails, which you can see here. This is a very big crab ecotype snail. And this is a very tiny, small ecotype here. And then the second axis is more correlated with the shape, with like very big aperture here. And this is the other extreme snails here. But generally speaking, this is only Spain as the flag shows. Generally speaking, you see that this is the phenotypical pattern is way more continuous than the genetic one. So we don't have those two clear clusters as we had in the genetic in the things, but we can still kind of clearly see a difference between the two ecotypes in that PCA. The other thing that we were interested in is to see if we can see also a phenotypic variation within the along the shore. And again, we found that pattern in the crab ecotype that look more and more wavy as we go down the shore, which is kind of an indication that potentially there is either like local adaptation in those or with introgression from the wave, or yeah, but we don't know exactly what's going on. And then this is just to show, this is the phenotypic component versus the genotypic component. So you can clearly see the two different clusters that are like, there's a quite good correspondence between genetic and phenotype. And yeah, so this didn't, so on average, the crab ecotypes, they are bigger, they are heavier, and they have thicker shells. And they are also shyer compared to the wave ecotype, which is also consistent with what we found in Sweden previously. Okay, so what we found in this study is like we have higher level of genetic differentiation in Spain over the whole genome. We also have this like two distinct cluster genetic pattern, even if we remove the inverted regions. We didn't find any hybrids in that study, though I can say that we are doing a follow-up study and we find some occasional hybrids, but they are quite rare compared to the Swedish system. We also have some level of parallelism in the sense that the same regions of the genome show very high differentiation in both systems, in both countries. And we also found the inversions in Spain. And this is very interesting, like sex determination loci inversion, on LG14. We also found a high level of phenotypic differentiation and a good correspondence between genotypic and phenotypic groups. So overall, like the Swedish system compared to the Spanish system. So the Spanish system represents a later stage or a closer points on the speciation continuum that is closer to be fully, that has to have completed speciation. So what can be the reasons for that? So, well, first of all, as I said, there's time. So it seems that the Swedish ecotypes are older than the Spanish are older than the Swedish ones, which probably this is because also they survived the Pleistocene while the Swedish are very recently colonized. And so this probably gave more time for local adaptation and other things to act on the system. The other thing is like this arrangement of the contact zone is a bit different because in Sweden, the contact zone is very, very narrow and you don't have, most of the snails cannot encounter the other ecotype most of the time, while in Spain, it's more likely to encounter. So that gives more opportunity for reinforcement. Another thing is that, so we can breed those snails and do some crazy like crossings between them. And so there've been crossings between Swedish ecotype and between Spanish ecotype, and there is a way higher abortion rate when you cross the crab ecotype and the wave ecotype in Sweden, and that creates a selective pressure for the reinforcement of pre-zygotic barriers to gene flow. That can be one thing. And then what I was mentioning a bit before is this selective gradients. So in Sweden, we have both, we have this like crop predation versus wave action gradient, but we don't really have a tide desiccation gradient, or at least it's perpendicular to the crab versus wave gradient. While in Spain, those two gradients are completely collinear. And so overall, it creates probably a stronger divergent selection pressure that could also explain part of the why there is a higher reproductive isolation in Spain. So with that, I want to thank all the co-authors of this paper, in particular Francesca here, because we were both co-lead author on this study, and I'm happy to take any question.